So all the efferent fibers passing the skeletal muscle are excitatory. Unlike your cardiac and smooth muscle, there you can have excitatory or inhibitory. But your glutamate is the primary neurotransmitter here. Each spinal motor neuron innervates only one kind of muscle fiber. We'll be talking about it. There are two kinds of muscle fiber. We have your fast fibers and we have your slow fibers. We'll be talking about the difference. Depending on the type of muscle fibers, they innervate motor units are of two types, slow and fast motor units. Recruitment of motor units at rest. So at rest, there is only pretty much spontaneous motor activity happening. That is some baseline spontaneous discharge which happens. Whereas minimal motor activity, some motor units are activated. Good voluntary effort, large amount of motor units are stimulated. What is the gradation of muscle response? So, if repeated, it just means that repeated stimulation recruits more and more motor units. That's what they mean by recruitment. And it is said that the duration of the stimulus is directly proportional to the amount of motor units that get stimulated to get the work done. Now, what are the types? We have the white muscle fibers and the red or slow muscle fibers. Red are your type 1, white are your type 2. So, simple common sense, when I say fast, this means that this is a lot of energy. This has a lot of glycogen capacity in the form of energy and ATPase activity to break that glycogen. And these are responsible, as the name says, for fast motor conduction activities. They're really rapid, and that is have 50 times the contractile force of the smaller units, therefore also called fast muscle fibers. So the transmission is fast, the contraction is high intensity, high energy, and the duration it lasts also fast. Because of this high energy, it is easily fatigable. It gets fatigued easy, and it is used only for high intensity workouts. Like you're running, this is the type of muscle fibers that come into play. And red, exactly the opposite. They have low glycogen, low ATPase. They are slow. They are slow to get the transmission. They are slow to act, but stays for long. That is, they are resistant to fatigue. They are highly vascular and abundant mitochondria. This is required in muscles which maintain posture. That is the muscles which constantly at play. You need red fibers. Whereas for fast and rapid action, like running, you need your marathon. Long distance running. So the proportion of the type 1 and type 2 fibers are genetically programmed. You can work out and try to focus on hypertrophy of these muscles, but the composition, it's all genetically programmed. Now, there are many sources of energy for muscle contraction, that is step by step. So muscle contraction requires energy, which is predominantly your ATP. Now, at rest, the energy used is the oxidation of stored fat in the form of free fatty acids in case of rest and light exercise. And in case of a little bit, if you increase the intensity, there is breakdown of glucose and glycogen. That is a store. There, then the glycogen is utilized. Glycolysis happens. And so in cases of stronger muscle contraction, there are three mechanisms that come into play. So in case of stronger muscle contraction, one, these are rich in, they need oxygen to function. So the oxygen supply is released from the myoglobin. But this happens only when there is an decreased oxygen tension in the muscle fiber. So these release oxygen release and causes oxidation of glycogen. Second is your anaerobic mechanism. Metabolism takes into place. Anaerobic glycolysis happens and lactic acid is formed. Third, if these are mechanisms, we have something called creatine phosphate. Phosphocreatine mediated by CPK. So if there is hardly any energy or like a very bad catabolic state, what happens is they give their phosphate molecule. ADP takes the phosphate from the creatine and phosphate and forms ATP and creatine. So, but once the energy levels are restored, that like in case of starvation, if you eat, and in case of hypoxia, if you have good oxygen, then the reversible of energy once is restored. the creatine phosphate is again formed. So that's about skeletal muscle. We'll be talking very briefly about smooth muscle and a reflex, stretch reflex. Now, what are the general features of your skeletal muscle? Smooth muscle. They are involuntary. They are unstriated, hence also called a plain muscle. They are smaller. They are spindle shaped with varying dimensions. And that is, see, if the smooth muscle is in the eyes, they are small. Whereas in uterus, they are long. In the gut, somewhere midway between the iris and the uterus. So the length varies. The dimension of they're all spindle shaped, but the dimension of each with respect to the position and place varies. They have less mitochondria and depends largely on glycolysis for its metabolic needs. These contractions, these are something that needs to be constantly present. For example, gut motility is always happening. So they need to be sustained for a longer duration. They can also. They are more variable and produce less tension. So hence energy expenditure is also less. So it can sustain for longer duration. So here we don't have actin and myosin. Instead of the Z lines I told you, we have something called dense bodies, and instead of actin, we have something called actin and filaments. And they also appear a little bit striated on a microscopic level, like these. These are randomly thick and thin actin and filaments which attach to the dense bodies here, which also give a very irregular appearance. They don't have troponin. This thing we already spoke about, the single unit and the multi-unit smooth muscle. The single unit, they are in large sheets, they are low resistance bridges and function in a syncytial way. They have gap junctions which transmit depolarization impulses from one to another and can travel long distances like that. So these are predominantly present in the GIT, bile tract, ureters, bronchi, uterus, urinary bladder, things like that. So the muscles are characterized by appearance of spontaneous activity in certain areas called pacemakers. So I told you. So again, coming to this, these are non-syncytial. These are individual components. They are discrete. And these are your iris, ciliary muscle, pilomotor muscle of skin, sweat glands, sweat and eye muscles. So what is here is these have richly innervated nerve supply. They need the nerve transmission for it to contract. Whereas this, like the cardiac muscle, have pacemaker tissue. So there are two types of spontaneous electrical activity that happens here we'll be talking about. Sometimes these pacemaker tissue or so this electrical activity, if it goes above the threshold, it has the capacity to generate an action potential or a spike potential on its own. It does not need a nerve innervation to do it. The nerve innervation here basically modulates the activity, but has nothing to do with muscle contraction here. In fact, they are constant at play. There is a constant there is a constant electrical activity and a baseline tone that is maintained in these GI muscles. So to say they are like almost at a contracted state. If these muscles are stretched, there is production of active tension. These muscles do not respond to stretch, but that's basically what's mentioned here. So we'll be talking about the electrical properties of the we'll be going to the diagram again later. Electrical properties characterized by the instability of the membrane. There is no specific membrane potential, like the nerve at -70, the muscle at the skeletal muscle at -90. There's nothing like that here. So it is very variable. The resting value being if the muscle is active, so the threshold is lowered. So there can be a lot of spike potential produced at that time. If the muscle is at rest, the threshold is a little higher and the spike potentials are not generated. It's in brief. Initiation of spontaneous activity, it begins with calcium influx. Like anywhere else, muscle contraction starts. The electrochemical activity that happens, the first chemical activity that happens is a calcium influx. They are superimposed on membrane potentials. Okay, these are the pacemaker waves I was telling about, the baseline waves. There are two types. One is your slow sine wave like fluctuations. The other one is a pacemaker potential. So not only do they have active action potentials, but they also have these baseline functions. So this is a baseline slow sine wave function. But if this does reach the threshold, it forms an action potential, like we see here. And this action potential can be present in the upslope or downslope. These pacemaker, on the other hand, as this potential can be stimulated in multiple sites, and they have the capacity to travel long distances also. That is a pacemaker potential. Now, this pacemaker potential, same thing, but only on an upslope it can form a spike potential. Both of this, it's understood that it has to cross the threshold to form an action potential. So like I said, so it makes sense. So you don't need like a nerve innervation for an action potential to be generated. Like spontaneously also, it's just happening. It just has to reach threshold. Now, mechanical properties, continuous irregular contractions that are independent of it or independent of its nerve supply is present. It maintains the state of partial contraction is called the tone, tonus or the tone. The muscle contraction, see the correlation between an action potential and muscle contraction is little, doesn't even correlate. Approximately, the muscle starts to contract approximately 200 milliseconds after the start of the spike. The peak contraction is approximately 500 milliseconds after the spike. Therefore, the excitation contraction process is very slow. Now we have something called the latch bridge mechanism. It can maintain prolonged tonic contraction for hours with little use of energy. The maximum force of contraction of smooth muscle is often greater than that of the skeletal muscle due to the prolonged period of attachment of the myosin cross bridges to the actin filament. Importance is visceral organs such as the GI tract, urinary bladder, gallbladder, etc., can maintain a tonic muscle contraction almost indefinitely. That is, the cross bridges that formed, they do not latch quickly and they also do not disconnect quickly. So it stays in a state of contraction for longer period of time. That's why it has the capacity to maintain this basic tone of the GI tract and the urinary bladder and the uterus. So unique feature of the visceral smooth muscle, in the absence of extrinsic nervous innervation, stretch induces contraction. The stretch is followed by a decline in membrane potential, followed by increase in spike frequency and hence the general tone. So, like if you're consuming food, there is a stretch of the muscle that itself can reduce the membrane potential, that is the threshold, so that these low sine waves or the pacemaker tissue can quickly create action potentials, contract, propel the foot forward. So that's what they mean. Even not only nerves, just the stretch can stimulate an action potential and cause contraction. That is present only in a visceral smooth muscle. So how the mechanism of contraction is a little different from a skeletal muscle here. Here something called a calcium calmodulin system comes into play. So we have ACH, which binds to muscarinic receptors. There it was nicotinic. Increased calcium influx into the cell, binding of calcium to calmodulin. This is the first. So calcium influx for contraction is it's ubiquitous. Once calcium enters, it binds to calmodulin. Now this activates something called the light chain kinase enzyme. And this causes phosphorylation of the myosin head. And apart from the phosphorylation, there is activation of the ATPase enzyme in the myosin head. It phosphorylates, it activates the enzyme. And because of the activation and also the site exposure during the same time because of the calcium, there is binding and the slide. Relaxation is the same thing as the calcium levels falls. There is dephosphorylation of the myosin head, and because of the dephosphorylation, there is dissociation of the calcium calmodulin system, and there is relaxation. Or there can be sustained contraction, which we just spoke about, the latch bridge mechanism, where the cross bridges can stay just like that and not detached for almost an hour. This maintains the basic tone of the gut. So length tension relationship is not like what we saw in a skeletal muscle. It's quite haphazard. So here they did an experiment with the urinary bladder. So they kept injecting 50 cc of fluids into the urinary bladder, and they found the tension that developed in the muscle. It was quite jagged. It would go up, it would go down, and they removed the same amount of fluid. they kept removing 50 cc after it reached the 700 cc. They kept removing 50 cc, but you see that they do not correspond. So this property is called plasticity. The smooth muscles are quite malleable. So it does not follow a specific law. Now, what is the nerve supply of the smooth muscle? We have autonomic nervous system. So this is, see, you can have the adrenergic nervous system, which can stimulate muscle contraction and cholinergic causing relaxation, or it can be the other way around. Cholinergic can cause stimulation and muscle contraction, and adrenergic can relax. So, but eventually the pathway is all the same from the spinal cord to the efferent preganglionic fibers. This is the efferent pathway I'm talking about. Efferent preganglionic to the ganglia, postganglionic fibers, and it runs along the length of the muscle. See, this is, it runs along the length of the muscles. These are the varicosities you see. They have chemical transmitters within them. Once they're stimulated, these are released intermittently, and these can respond even to stretch. So, but this does not go too far. It restricts. So, for example, if there's a release, it restricts itself to the close by sensation. Like it does not cross beyond one filament to the other, because it does not pass through the gap junction. But if there is repeated stimulation and repeated stretch and the amount of transmitter keeps on increasing progressively, then the whole muscle fiber can be stimulated. Okay. Release activates muscle fibers only after the sensation. Therefore, single stimulation does not cause contraction, whereas repeated stimulation can cause. This is the varicosities I was talking about. These are vesicles with neurotransmitters. They release. This is a muscle fiber. They have receptors for it. Now we have same concept again. We have something called excitatory junction potential. That was at the level of the fiber membrane. This is at the level of the junction. So in smooth muscle with excitatory adrenergic discharge, stimulation of adrenergic nerve causes discrete partial depolarization. See, these are not enough to create a spike potential, but on repeated stimuli or on summation, it can produce a full blown depolarization. Now we have ions. Will be ions and catecholamines and ACH and their effect on the membrane. So effect of catecholamines, we know they act through beta and they act through alpha receptors. Now via alpha, what it does is efflux. There is a calcium efflux. And via beta, it stimulates the CAMP and increase the intracellular binding. So there is no free calcium here for contraction. One, the calcium from ICF is going out, or whatever free calcium is spent, it is bound. So as a result, hyperpolarization occurs. There is an increase in membrane potential, threshold potential. So because of which you need the current that comes in to be a little more higher to cross the threshold to form spike potential. Not only that, not only the membrane potential is affected, the baseline spike frequency is also reduced. So hence the baseline tone of the muscle is also reduced. So one, the threshold is increased, and the other one, the frequency of spikes also, eventually it causes muscle relaxation. Catecholamines cause muscle relaxation through all mechanisms. ACH does the exact opposite. It causes influx, it causes depolarization, it reduces the membrane potential, increases the spike potential, causes depolarization. Now coming to ions, calcium, it's explained enough. High calcium in the ESF, increased spontaneous muscle contraction. High calcium in the ESF causes sodium influx. That causes depolarization. Sodium as such has no effect. Calcium leads to sodium influx. Barium, direct smooth muscle stimulant, marked increase in frequency and tone of force of contraction. It acts like an irritant. Now that's about smooth muscle. It's already 9:15. Now this is the monosynaptic. We'll be talking about Golgi tendon body and nuclear bag. And next class. Okay. Manjunath, sir. There's probably not, I think.
Can I hear?
Sir, shall I do the monosynaptic reflex and Golgi tendon body for a later date? It's already 9:15, sir.
Yeah, it's already 9:15.
Yes, sir.
Next
Okay.
one more session you can keep it.
Okay, sir. Okay, sir.
Thank you.
Thank you, sir.
Covered very well.
Thank you, sir.
Covered.
Thank you, sir.
Yeah.